Well, thank you very much for the invitation to come speak at this wonderful meeting. It's really great to hear such a delightful mix of clinical and basic and applied science. So I changed my title a little bit to make it a bit more general: transcriptional regulation of heart development. This is my disclosure slide. And so what I want to tell you about is two vignettes of approaches that we're using to try to understand how nature uses its own cues to make heart cells, and how that goes wrong in the case of disease. And what I've illustrated on this cartoon here is basically the general theme of what we're trying to understand, which is how does an uncommitted precursor cell, so an embryonic stem cell, for example, acquire a specific lineage and then differentiate into specific cell type. And so with the same genome at play, how do these cardiac myocytes up top acquire an identity and a function that is considerably different from these neurons at the bottom, again with the same genome as a template? And so the biology that we interrogate is gene regulation. And here, this is a cartoon of basically a variety of different mechanisms that are involved in gene regulation, in turning on or off genes during differentiation. And in the last several years, we've addressed pretty much all of these different mechanisms in certain ways. And what I'll tell you in the first part of the talk is some of the work that we're doing on trying to understand how DNA-binding transcription factors, these proteins that recognize a sequence motif on DNA, are involved in regulating cardiac development, and how they're involved in human congenital heart disease. So the protein that I'm going to tell you about is called TBX V, T-box transcription factor 5. It's a DNA-binding transcription factor of the large T-box family. And this was discovered in 1997 in John and Cricket Seidman's lab as the first gene to be associated with congenital heart disease as part of a rare syndrome called Holt-Oram syndrome. The cardiac manifestation of Holt-Oram syndrome is shown below. This is my only clinical slide, where the patients have ventricular or atrial septation defects as well as conduction system defects. And in the top right is expression of TBX5 in mouse embryo, showing that it's expressed throughout cardiac embryogenesis, starting very early on, and acquires later patterned expression. Oh, I should say, two things that are important here is that this also, this discovery in humans told us that developmentally important gene regulators were likely to be at the root cause of congenital heart disease, so malformations of the heart, and this has been borne out by other mutations. The other thing to note is that the mutations that cause these defects are heterozygous mutations. So there's only one copy of the gene that's affected. That implies that the level, the dosage of the transcription factor is really, really important. So bear that in mind. Okay, one thing that I discovered that's intrigued me for a long, long time is this pattern of gene expression. So blue is the RNA for TBX5 in a mouse embryonic heart at around embryonic day 11.5. You can see the two atria and the two ventricles. And what you can see is that TBX5 expression is high in both atria, but only in the left ventricle and not in the right ventricle. And if we were to section this heart, what's shown in cartoon form on the right, is that there's a boundary of expression of TBX5 that bisects the interventricular septum. And this, we think, can explain how reduced dosage of TBX5 can cause ventricular septation defects by disrupting this boundary. Okay, and this we know from mouse experiments, but we'd like to understand human congenital heart disease or human disease. And fortunately, we can take human iPS cells and turn them into beating cardiac myocytes, and we can engineer them, and I'll show you some of our efforts related to that. But I would challenge the most astute cardiac anatomist in the room to point to me where the interventricular septum is in this field of cells, or where the atria are, or where the ventricles are. It's a two-dimensional sheet of cells, and how could we possibly model congenital heart disease? Well, I'm going to show you that we're trying, and we may have some success in this. Okay, so we took the approach we took was not to make patient-specific iPS cells, but to take a well-characterized iPS cell line that was developed in Bruce Conklin's lab, my lab and office neighbor, called WTC11. And we engineered in using CRISPR-Cas9 editing either a heterozygous or a homozygous loss of function. I don't know if I can point. Here we go. Heterozygous or a compound heterozygous, but for all intents and purposes, homozygous loss of function allele of TBX V. And this leads to parental or control cells that are controls. The control has seen the CRISPR-Cas machinery, but does not have an indel. And then we have heterozygous lines that have half the amount of TBX5, and then homozygous lines that don't have any TBX5 at all. The first thing that we did was to look at to see whether these lines beat. The control lines and heterozygous lines have about the same onset of beating and the same differentiation efficiency, so that's good. The homozygous lines are delayed in their onset of beating and have less differentiation efficiency, consistent with the role for TBX5 in driving cardiac differentiation. By electrophysiology, using either patch clamp or fluovolt, we were able to find that we had gradually prolonged action potentials in the heterozygous and then more pronounced in the homozygous, and this is quantitated here. So there's only a trend in the heterozygous by patch clamp, but there's a overall significant difference by fluovolt. And so we have these electrophysiological defects that are consistent with the human disease phenotype. So in a sense, this is satisfying because we can tell that there are cellular defects. But of course, we're again have these cells in a dish. Can we learn something about the different cells that might inform the structural defects that arise from these mutations? Oh, they also have other things like EADs. Apologies. So to look at gene expression, which is sort of what we want to understand downstream of a transcription factor, we would normally do bulk RNA sequencing to look at changes in gene expression. But again, by taking the entire dish, we would only see the most pronounced changes in gene expression that we might expect. So instead, we turned to single-cell RNA sequencing using the 10x Genomics DropSeq platform that's illustrated here. And what this does is that it takes barcoded gel beads, thousands of barcoded gel beads, and then using a microfluidics device, cells and enzyme are brought in with the beads and encapsulated within a lipid microdroplet. And then these droplets basically isolate the cells, and within these droplets one can do the sequencing, the reverse transcriptase and sequencing reaction for thousands of cells at once, and then each cDNA is barcoded, thrown onto a sequencer, and so then one can obtain the transcriptomes of thousands of individual cells. And so the data look like this. This looks like a big messy cloud, but it's actually a two-dimensional statistical representation of the transcriptomes of over 13,000 cells. And so what this is it's called a t-SNE plot. And it's using multi-dimensional principal components analysis for these thousands of transcripts and thousands of cells, and then compressing it into 2-dimension. And what you can see here that they're colored are groups of cells. So each dot represents the transcriptome of a single cell. And so there's 13,658 cells here. And what the statistics are doing is grouping together cells that resemble one another based on their gene expression patterns. So these cells in this orange group here, group 1, are more related to one another based on their gene expression patterns than they are to these cells in group 2 and group 3. Okay, so we have these 29 different groups of cells, some of them very large, some of them very small, and we can ask the software to tell us which ones correspond to which genotype. And so you can see here in black are the controls, in red are the TBX5 heterozygotes, and in blue are the TBX5 homozygotes. And so you can see right away that we can discern not only different types of cells based on their single-cell RNA-seq, but also groups of cells that segregate based on the different genotypes. Okay, so what can we learn from looking at these plots? Well, first, we can understand the diversity of cell types that we have in our dish. And so here we've asked the software to paint cardiac troponin T over the clusters. As you can see, there's a very large cloud of cardiac myocytes. And that's not surprising because we're trying to make cardiac myocytes in a dish with a protocol. And then there's these groups of cells here that express some degree of troponin T, but less so. And over here on the right is KDR, a marker of endothelial cells. And you can see that we have a large group of endothelial-enriched cells over here, and then this tiny, tiny little Y-shaped group of cells that we think is vascular endothelium based on its signature. So two things to take away from here. We can look at the diversity of cell types that exist within the dish, but also we can identify very rare populations. This corresponds to under 1% of the cells that are in the dish. We never would have known that these cells exist had we mushed up the entire dish. Okay, so what can we learn about the genotypes? So we focused on these three clusters here that segregate very clearly based on genotype, either wild type, heterozygous, or homozygous. And we asked the software to tell us what's different between these clusters. One gene that stood out is probably one of my favorite genes of all time that I worked on even as a graduate student, NPPA, encoding atrial natriuretic factor. And you can see that it's enriched in the control cells, but it's reduced or absent in both the TBX V heterozygous and homozygous cells. So here's a gene that's very sensitive to TBX V dosage, where even with 50% drop of TBX V, its expression is reduced. This was very satisfying to me because at the end of my postdoc, I published in a mouse model of TBX5 deficiency that this exact gene, A and F, was sensitive to TBX5 dosage. So we're recapitulating in this 2D human iPS cell model what we knew already in the mouse. So that's great. We can also discover new things. For example, an overexpression in some of the heterozygotes and in all the homozygotes of a gene called R-spondin3 that normally is not expressed in the cardiac myocytes. And there's hundreds of genes that we're now exploring and trying to understand what really is the molecular etiology of this disease. And we can quantitate this, I just want to bring your attention to why some of these genes are important. Well, R-spondin3 is a secreted coactivator of Wnt signaling, and we know that overexpression in cardiac precursors causes heart defects, so this might be something important. And then we also, another gene that I didn't show you the feature plot for is HAND1, and we know that HAND1 misexpression leads to a single ventricle in the mouse. So we think now using this human model, we're really getting at, even though it's a 2D system, getting at understanding how these congenital heart disease-inducing mutations are involved in broadly organizing gene regulatory networks that might be at the root of these defects. Okay, so now I want to move more briefly to the second part, which has to do with something that interacts with the DNA-binding transcription factors, which are the chromatin remodeling complexes. So these are large complexes that come in and move around nucleosomes in the genome, organizing the structure of genome to open up or close up chromatin so that the DNA-binding transcription factors and other things can access or be kept away from gene regulatory elements or from promoters. And a family of chromatin remodelers that we've been studying for a number of years is the so-called BAF chromatin remodeling complex, this big ball that I'm showing you here. And all the different sort of names of the different components aren't so important right now, but just to keep in mind that this is a multimeric assembly of a variety of proteins that can be encoded by different flavors. So there's many different forms of these complexes, and we're trying to understand the significance of its assembly. But what's circled down here is the main engine of the complex, the ATPase, the actual machinery that unwinds the DNA. And there's two proteins that are represented that are mutually exclusive in the complexes. One is called BRG1, and Brahma-related gene 1, and the other is called Brahma. And we know that BRG1 is very important for pluripotency, it's important for mesoderm induction, but it's less and less important in gene regulation as differentiation proceeds. And so the question in the field has been, well, what about Brahma? What is Brahma important for? What's this other ATPase important for? And it had been thought, based on mouse knockouts, that it's dispensable for development. Well, we, for a variety of reasons, including the ease of genome editing with CRISPR-Cas, went ahead and made Brahma knockout embryonic stem cells and asked, do they still make cardiac cells? And so these are 3 movies that are ongoing, and you can see that. So the wild type line is on the left. It's exuberantly beating away, and the 2 Brahma knockout lines that are on the right are not beating at all. This is a movie. They're not beating at all, at all, at all. So what's going on? Are they making heart cells that don't have function? We looked by troponin T staining and by gene expression. And on the left is troponin T staining, where in green we have the 90% of cardiac myocytes that we normally see in a wild-type culture. And this down here, I think in red, is the knockouts. There's only maybe 2% troponin T positive cells that we can measure. So basically, there's no cardiac myocytes in there. But there are cells. And so we did RNA sequencing. And here's a heat map showing you in red genes that go up and in blue genes that go down. And what we find, and this is at a cardiac precursor stage and at a cardiac myocyte stage, where normally you'd have cardiac myocytes. And we find that early on there's a failure to activate some of the important cardiac transcription factors, but not complete failure. Later on, there's an absolute complete loss of any expression of the contractile protein genes. So all the contractile protein genes shut off. And instead what we get is we get genes that belong in other cell types, in other mesoderm, and even neural genes. So we wondered, well, what's going on? Are the cells having a confused identity where basically now the genome is drunk and just turning genes on randomly? Or do we have a weird mix of cell types? And so again, we turn to single-cell RNA sequencing to determine what kind of cell types we have. And so here's the t-SNE plot of about 8,000 cells in blue and green at the cardiac precursor stage, and in red and purple at day 10, where cells normally make cardiac myocytes. And we have the wild-type cells are in blue or in red, depending on the stage, and the Brahma knockout cells are in green or in purple. And what you may appreciate is that there's not much difference statistically between the Brahma knockout cells and the wild-type cells at the precursor stage. These 2 clusters are defined by very, very minor changes in gene expression. But there's a very broad difference, at least statistically, between the Brahma knockout cells here in purple and the wild-type cells. So what are these Brahma knockout cells? It turns out that, so here I've painted the same t-SNE plot with troponin T in the wild types, and then with a variety of different markers that light up the various knockout clusters. And what we find is that instead of cardiac myocytes, what we're getting is osteoblast cells. So that's okay, that's a mesodermal derivatives. But we're getting brain cells, we're getting retinal neurons, we're getting oligodendrocytes, a complete shift in cell identity in the absence of Brahma. So we're making cardiac mesoderm, we're getting to a cardiac precursor stage, and then all of a sudden the cells are completely losing their identity and making cells that actually come from different germ layers, from ectodermal germ layers. And so we're now in the process of trying to understand this. We've looked at chromatin, the openness of chromatin using a technique called ATAC-seq, and we find that the absence of Brahma is closing up some of the cardiac enhancers, and it's opening up enhancers that belong to these other cells. And so what we do know, and what I'll conclude with, is that Brahma is absolutely essential for the transition of a cardiac precursor to a cardiac myocyte, but is important more than just for allowing this transition. It's important as a safeguard for what turns out to be a very fragile cellular state, and that in its absence, the cells will undergo complete fate shifts into cells that normally would never come out of cardiac precursors. So what does this mean? What do we take from this? It means that when we think of cardiac differentiation, if we think of cardiac disease and a reversion to a more primitive state, we can think of this as an extreme example of such a reversion or diversion of states. And it also perhaps informs us when we think about strategies to regenerate the heart, to create new myocytes, that maybe we'd have clues here about what the important safeguards we might want to put into place when we're making new cardiac myocytes in regeneration. So I'll close there and thank the folks in the lab who did the work. The TBX V work was done by Irfan Kataria, who's here, who's a pediatric cardiac anesthesiologist in the lab. And the work on Brahma has been done here by Swetansu Hota, a postdoc in the lab. And I'll thank you very much for your attention.